didn't expect you to be early. Please, make yourself comfortable and take a seat. My name is Noah Linsky, and I'm a biology and Spanish...
Take two.
As I was saying, my name is Noah Linsky, and I'm a biology and Spanish minor languages major with a minor in chemistry. Starting last August, I had the remarkable opportunity to study abroad in Alicante, Spain, a small city on the southeastern coast of the country within the community of Valencia. I lived in Alicante for almost 8 months and had the privilege of working in a biochemistry laboratory for the second half of my study abroad experience until I was advised to return home due to COVID-19. I will be presenting my research findings at the IGP Summit on the 9th of November. Below, I'm excited to include a summary of my research abroad, along with how this research is biologically and environmentally relevant. I performed my research at the University of Alicante, located about an hour away from the shore by tram. My laboratory investigated amino acid synthesis in the nitrogen cycle within the species of Archaea, Halophilic Archaea. The nitrogen cycle is a series of processes by which nitrogenous compounds are exchanged between certain species and the environment. Archaea are microscopic, single-celled organisms that often live in extreme areas of high salt concentration, high temperature, and even high acidity. These organisms, like all life forms, create amino acids, the building blocks of proteins, with help from their personal contribution to the nitrogen cycle. This species is predominantly found living in stagnant pools of very high saline concentration along the Mediterranean, and therefore needs an acidic environment in order to survive. Halophilic Archaea participates in the nitrogen cycle through the assimilation of nitrogen, which is the conversion of nitrate, NO3, into ammonia, NH4. With the help of specific enzymes and natural energy sources, this species uses the assimilation of nitrogen in order to create amino acids. Enzymes are proteins that have the ability to speed up chemical reactions and are essential to the continued existence of all organisms, even you. My team and I specifically studied the synthesis of glutamine, a polar amino acid with an uncharged side chain that can be incorporated into a variety of metabolically necessary proteins. Using various forms of biotechnology, my laboratory researched the function of 2 specific genes, glutamine-A2 and glutamine-A3, present within the enzyme glutamine synthetase. Genes are segments of DNA that include the synthesis of proteins. This enzyme, glutamine synthetase, has the ability to catalyze the breakdown of the compound glutamate along with ammonia from the nitrogen cycle in order to create the essential amino acid glutamine. This research not only gives us better insight regarding the mechanisms of amino acid synthesis, it also expands our understanding of the nitrogen cycle in species of Halophilic Archaea, which could lead to future advancements in bioremediation, which employs the use of living organisms in order to clean up contaminated areas. Now, thank you all for listening. Given your extensive familiarity with halophilic microbiology, I would definitely recommend this presentation to you and anyone else you know. Just kidding. You don't need to know anything at all about microbiology, and you still will have a wonderfully enlightening time. I hope to see you all at the IGP Summit on the 9th of November, and I'm excited for all of the IGP students who are ready to go abroad. Thank you again for your time. Now please, if you could excuse us, my friend and I have plenty of work to do.